The plasma membrane is a membrane of lipids and proteins that forms the external boundary of a cell. It is also called the lipid bilayer. It is called this because it is made up of two opposed layers of phospholipids. Its purpose is to keep a barrier between the inside of the cell and the outside. It achieves its function because of its structural components. The first component is the phospholipid. This lipid is composed of a head and a tail. The head is hydrophilic and the tail is hydrophobic. The combination of these properties makes the membrane amphipathic. Many of these phospholipids come together to form the membrane. The hydrophobic tails pack together away from water tail to tail, and the hydrophobic heads face the watery environment both inside and outside the cell. Lipid bilayers are also self-sealing. Because of this property, they easily form a liposome, but it's actually just a cell. The lipid bilayer chemistry causes them to spontaneously roll into balls to conserve energy. This is crucial to the cell because it does not cost the cell energy to create the shape of the cell membrane. The lipid bilayer is also asymmetrical. This means it is not even on both sides. This is an important property because it means there is an outside layer and an inside layer. One is supposed to face the extracellular environment, and the other faces the intracellular environment. The distinction is made by how the membrane proteins fit. They have to be facing the correct direction. The fluidity of the lipid bilayer depends on its composition. More fluid membranes are less dense and easier to float in. Less fluid membranes are denser and harder to get around in. A contributing factor to how fluid a membrane is how closely the membrane lipids pack together. The tighter the packing, the denser the membrane. The packing also depends on the hydrocarbon tail length and the number of double bonds in the tails, known as kinks. Animal cells regulate membrane fluidity using cholesterol. Cholesterol fills in the gaps between kinked membrane lipids, creating a stiffer, less fluid membrane. Below the membrane is the cell cortex. The cell cortex keeps the shape of the cell. It is made of long tubular proteins that attach to transmembrane proteins to connect the cell membrane itself. The cell membrane also has a carbohydrate layer of the exterior of the cell. Many of the lipids on the exterior of the lipid bilayer are glycolipids. They have sugars attached to their heads. These sugars are large complex sugars made of multiple individual sugars linked together called oligosaccharides. This carbohydrate layer provides protection against mechanical or chemical damage. Specialized proteins called lectins recognize and bind tightly to individual sugars on the lipids of this layer. This mechanism is important for cellular recognition and cell-to-cell adhesion. Membrane proteins have many different functions. A few of their functions are to transport molecules that would not normally be able to move across a membrane or anchor proteins as receptors. Integral proteins are proteins that are permanently attached to the cellular membrane. Another name for them is embedded proteins. Transmembrane proteins are one type of integral protein. These proteins extend completely through the membrane with hydrophilic regions on either end. They can covalently link to a phospholipid head or non-covalently associate with another transmembrane protein. They are in a fixed position within the membrane. Peripheral proteins are associated with the cell membrane, but they do not enter the hydrophobic space within the cell membrane. They associate with the hydrophilic heads of the membrane. Transport proteins are a type of membrane protein. They are found in all cell membranes. Transport proteins are exclusive, meaning they are specialized and only allow transport for a single or very limited number of related molecules. The types of transport proteins are transporters and channels. To understand how transporters and channels work, we have to know what a concentration gradient is. A concentration gradient means that the concentration of the outside of the cell is different than the concentration in the inside of the cell. This makes it so that molecules want to move in and out of the cell to reach equilibrium. The molecules can be motivated either electrically or chemically. This is called an electrochemical gradient. For example, if we have all of these blue molecules on the inside of the cell and only a few on the outside, they experience a chemical pull to move to the outside. If we have a lot of negative molecules on the inside of the cell and a lot of positive on the outside, they feel a pull to switch the side of the membrane they are on. These concentrations are crucial for the cell's survival. The inside of a normal cell is usually negative, and the outside is usually positive. This charge difference is known as the resting membrane potential. It means nothing is occurring in the cell, and it has no stimulus. At this time, ions and molecules want to move, but cannot. A transporter is a protein that contains moving parts and can transfer molecules from one side of the membrane to the other. They often move molecules against their concentration gradient, which requires energy. A transporter needs to be activated by the molecule binding to it. This causes a shape change, which lets the binding signal through. A channel is a protein as well, but it does not usually have moving parts. These channels usually have substances follow their concentration gradient, which does not require energy. Channels are hydrophilic holes in the membrane that molecules can easily pass through. When molecules move through a transporter against the concentration gradient, it is active transport, which does require energy. Transporters that move molecules against the concentration gradient are called pumps. An example of this is the sodium-potassium pump. Based on environmental cues, the cell pumps sodium and potassium in and out of the cell. When molecules move through transporters or channels following their concentration gradient, it is facilitated diffusion or passive transport. This does not require energy. There are three main strategies for transporters to actively transport molecules against a concentration gradient. The first is classified as coupled transporters. They use one molecule moving with its gradient to power the movement of another against its gradient. The first type is a symporter. Molecules moving through symporters are moving in the same direction, with one molecule moving against the gradient from low to high, and one molecule moving with the gradient from high to low. The second type of coupled transporter is an antiporter. Molecules moving through antiporters are moving in opposite directions, but still with one molecule moving with the concentration gradient and one molecule moving against. The second type of transporter protein is an ATP-driven pump. These use energy obtained from the hydrolysis of ATP to force the movement of molecules against gradients. The third is a light-driven pump, where a molecule absorbs photons and changes its shape to cause another shape change of the pump that allows the molecule to be squeezed through. Ion channels are specifically for small inorganic ions such as sodium, potassium, chlorine, and calcium. There are 4 types of ion channels. The first is a voltage-gated. The opening of this channel is blocked by a voltage sensor. This part of the protein is sensitive to changes in the membrane potential. An example of a voltage channel is on a neuron. If we have a cell at the resting potential, the voltage-gated ion channels are closed. When the membrane is depolarized, the conformational changes will open the gate. The second type of ion channel is ligand-gated. This channel is only opened when a specific molecule is bound to it. An example of this type of channel would be a synapse on a neuron. Neurotransmitters land on receptors on the channel to tell it to open. The third type of ion channel is mechanical. This type of channel is opened by physical stress, such as pressure. An example of this could be a skin cell. Their receptors react to pressure, which opens the channel. The fourth channel is always open for facilitated diffusion to occur. Vesicles are a very important component of the cell system. They are used to transport molecules from one part of the cell to the other. The vesicles are hollow balls of membrane that carry hydrophilic proteins and membrane components. Clathrin is a protein that coats the vesicle like a cage. This coat is important for 3 reasons. It helps to form the bud, it holds the shape of the vesicle, and it ensures the vesicle has the proper contents. The formation of a clathrin-coated vesicle starts at the cell membrane with the cargo molecules and receptors. The cell first recruits the AP2 complex or adaptins. This association of adaptin and the cargo receptor is what allows the clathrin proteins to bind. With the addition of the clathrin, an invagination in the membrane occurs. This forms the clathrin-coated pit. It is a basket-like structure with a clathrin mesh at its base. This is what begins the shaping of the vesicle. Once the pit is formed, a protein called dynamin forms around the neck of the vesicle like a collar. Its purpose is to pinch off the rest of the membrane, releasing the vesicle from the cell membrane. For uncoating, the adaptins and clathrin proteins are removed, leaving a naked vesicle. Docking occurs after vesicle formation. This is when the cell needs to find a way to its destination with its contents. On the surface of the vesicle are membrane proteins called Rab proteins, as well as a V-snare. The Rab proteins give clues to the cell about the vesicle's origin and what it is carrying. The target membrane has tethering proteins and a T-snare. The vesicle arrives at the target membrane. Now, the Rab proteins are recognized by tethering proteins. Tethering proteins are found on the destination membrane or where the vesicle wants to dock. The Rab proteins associate with the tethering proteins, and the vesicle gets pulled toward the target membrane. Next, further stabilization of the vesicle occurs through snares. Snares are proteins that provide a second layer of anchoring in the docking process. On the vesicle membrane is the V-snare, and on the target membrane is the T-snare. These snares are complementary and their linkage results in complete docking of the vesicle on the target membrane. Fusion occurs just after docking. 2 things result from vesicle fusion. The vesicle membrane becomes the target membrane, and the contents of the vesicle are released. So the cell arrives at the target membrane and the T and V snares associate, successfully docking. The closelessness that needs to be achieved for fusion to occur is caused by the snares as well. They pull the vesicle toward the membrane by twisting and winching so they are as close as possible. The vesicle membrane becomes the target membrane, and the membrane-bound proteins will be delivered into the cell. The cytoskeleton is a complex dynamic network of filaments found in the cytoplasm of cells. They have 3 main components: microtubules, microfilaments, and intermediate filaments, each with a different purpose for the structure of the cell. Microtubules are long, stiff, hollow tubes of proteins. They are made up of a subunit protein called tubulin. The tubulin proteins are dimers of 2 globular proteins. Each dimer has an alpha subunit and a beta subunit. The microtubule is formed when the tubulin molecules stack together with alternating subunits. When these subunits are stacked, they form what is called a protofilament. 13 of these protofilaments make a microtubule. They interact through non-covalent forces to form the tube. This formation means that microtubules have a beginning and an end. They grow out of and originate from a small structure near the center of the cell called the centrosome. Each protofilament faces the same direction, giving the microtubule polarity. The end of the microtubule linked to the centrosome is the minus end, and the free end is the plus end. The polarity of the microtubule is what gives them their direction. Without it, vesicles would not reach their destinations using them. The formation of microtubules is dependent on the centrosome. As mentioned, the centrosome is where the MTs originate from. They serve as tubulin organizers. The centrosome contains a third type of tubulin called gamma. This is a ring-shaped structure that serves as the nucleation site. It's the starting point for microtubule formation. Tubulin dimers are added to the positive end of the MT fiber via rapid polymerization. This occurs when GTP is bound to the dimer. When the dimer is bound to GDP, it causes depolymerization and the MT shrinks backward, sometimes all the way to the centrosome. This is a representation of dynamic instability. The MT can form and reform where it needs to, depending on GDP and GTP binding proteins. In order to keep the MT from falling apart, it needs to be anchored at its plus end. MTs form all the time and in many different directions. Sometimes they will find a place for their plus end and become anchored, and other times they won't and will disassemble. Microfilaments are also known as actin filaments. Actin filaments are thinner and shorter than MTs, making them highly flexible. They are only 7 nanometers in diameter, and each is a twisted chain of identical globular actin subunits. They all have polarity pointing in the same direction. There is a plus end and a minus end of an actin filament, just like microtubules. They usually join together with other actin filaments and create a bundle. Actin filaments can grow on either end, but it is easier and faster from the plus end. Actin filament formation starts when a free actin monomer is bound to ATP. They then bind with other monomers to form an actin oligomer. The oligomers form because they can be more easily incorporated into a growing filament. Once the filament is made, the ATP is cleaved to ADP and it reduces the strength of the binding for the monomers in the filament. This promotes depolarization. This means actin filaments are dynamic just like microtubules. This property costs a lot of energy. Because of this, they usually only persist for a few minutes after formation. In a cell, there is a high concentration of actin monomers, so to prevent formation of the filaments, accessory proteins are bound to the free monomers. There are also actin bundling proteins which hold actin filaments together in parallel bundles. They hold actin into the meshwork of the cell cortex. And there are filament severing proteins which cut actin filaments into small pieces, converting the actin into a more fluid, gel-like state. The main purpose of the intermediate filament is tensile strength. They help the cell withstand mechanical stresses like stretching or squeezing. There are 4 main classes of intermediate filaments. Keratin filaments are the broadest type found in the cytoplasm of cells. Each keratin is made up of a mixture of different subunits. This allows them to become very diverse in function and shape. The vimentin, which are found in connective tissue cells, muscle cells, and support cells of the nervous system. Neural filaments, which are found in neurons, and nuclear lamins, which are found in the nuclear envelope. The structure of the first 3 classes of intermediate filaments is rope-like. It starts with pairs of IF subunit proteins, or monomers, that wrap around each other to form dimers. 2 dimers then come together to form a staggered tetramer. The tetramers then bind to form a tetramer sheet. The sheet then rolls and forms a bundle of twisted fibrous strands. The lamins form a 2-dimensional mesh. They are different from the other intermediate filaments because they need to assemble and disassemble regularly. This is controlled by phosphorylation. The phosphorylation of lamins by a kinase weakens their affinity for other lamins, causing disassembly. Dephosphorylation by a phosphatase causes them to reassemble. Intermediate filaments can also be reinforced and stabilized by accessory proteins. One protein that does this is plectin. Plectin cross-links intermediate filament bundles into stronger arrays. They also link intermediate filaments to the other components of the cytoskeleton, providing strength against mechanical stress. Cell crawling is when cells sense their environment using receptors and migrate toward the signals using actin. The first step to cell crawling is anchoring. So we have a cell that is on the membrane and wants to crawl. First, sheet-like lamellipodia and filopodia are pushed out of the leading edge of the cell. They contain a dense meshwork of actin. The actin is oriented so that most of the filaments have their plus end close to the plasma membrane. The smaller and thinner protrusions are the filopodia. Each has a bundle of 10 to 20 actin filaments. The filopodia and lamellipodia are being sent out of the cell until they sense the chemoreceptor on the crawlable surface. Integrins are transmembrane proteins that bridge actin filaments to the surface on which a cell is crawling. So the integrins come into contact with ECM ligands and cluster in the cell membrane. This is the beginning of cell crawling via anchoring. All the cues that affect the functionality of actin for pulling culminate in a GTP binding protein called RHO. RHO triggers actin-myosin interactions to promote cell crawling. Actin polymerization occurs and lamellipodia and filopodia form. When RHO is active, the cell will crawl, and if it is not activated, the cell will not crawl. The next step is the cell pulling itself by contraction. But before we get to the contraction part of cell crawling, we need to understand muscle structure. The muscle structure starts with a muscle fiber, and within it are myofibrils, and the smallest portion of muscle is the sarcomere, and it has thin and thick filaments. The thin filaments are actin, and the thick filaments are myosin. Myosin 2 is the major myosin protein found in muscle. These proteins are two single myosin monomers twisted and bundled together. Thick filaments are the myosin 2 and 1 molecules self-assembled into thick bipolar bundles with head domains on each side. The actin runs parallel to the myosin. There are about 500 head domains on each filament, and they are each associated with 2 actin filaments. Contraction occurs through the sliding filament model. The thin filaments slide along the thick. So it starts with the myosin and the actin of a sarcomere, and troponin and tropomyosin bound to actin. At this time, the muscle is relaxed. The myosin head is bound to ATP, causing it to have a weak affinity for actin, and the arm lays flat against the thick filament. The tropomyosin are in a position where they block the receptors on the actin for myosin. When calcium binds to troponin, it changes the conformation of tropomyosin, revealing the receptors on actin. Then ATP is hydrolyzed, and the head domain has a high affinity for actin once again, and it binds. And the energy from the ATP hydrolysis causes the myosin head to pull the actin, while ADP and phosphate are released. Then another ATP binds to myosin and the cycle starts again. This is the release-bind-pull of muscle contraction. Contraction of muscle is an ATP-dependent mechanism. Now, if we look back at our original model, we have the anchored cell via actin and integrin interactions. On the other end of the cell, there is a contraction. That, combined with the polymerization of actin in the cell, it moves along the cell membrane. G-protein coupled receptors are the largest family of cell surface receptor proteins. They are sensitive to many signals, including but not limited to hormones, paracrine molecules, and neurotransmitters. They play a role in every system of cell communication. Specifically, GPCRs are transmembrane proteins found at the cell surface. They were synthesized at the rough ER and threaded through the ER membrane using start and stop sequences on an amino acid chain. Inactive GPCR sits at the cell membrane. It does not have a signal molecule in it. The G protein is made up of 3 subunits, alpha, beta, and gamma. These subunits bind to molecules that determine whether it is inactive or active. When inactive, the alpha subunit binds to GDP. When this occurs, the G protein is dormant and it is not doing anything. To activate the G protein, the first thing that happens is a signal molecule binds to the GPCR receptor. This changes the shape of the GPCR, which in turn activates a G protein that is associated with the receptor at the cytosolic face of the membrane. The change in shape by the receptor causes a secondary change in shape in the G protein. The change in shape of the G protein decreases its affinity to GDP and increases its affinity to GTP. This is important because G proteins bound to GTP are active. The G protein lets go of its bound GDP and binds to GTP instead. Once activated, the alpha subunit can alter target molecules and the remaining beta-gamma complex can do the same independently. This initiates a signal cascade that flows into the cell. The G protein also has GTPase activity. This means it can hydrolyze GTP to GDP. This causes the G protein to become inactive again and basically wait to be activated again. CAMP is a small molecule that is involved with G proteins and activation of certain enzymes. CAMP is known as a second messenger. This means it carries messages into the cell but does not carry any information itself. So we start off with a GPCR that has been activated by a signal molecule, resulting in the change in shape of the G protein that is bound to GTP. The alpha subunit of the G protein binds to adenylate cyclase, which activates the enzyme. Adenylate cyclase then converts ATP into cyclic AMP. CAMP activates another protein called kinase A. This is what will achieve a physiological response. Protein kinase A phosphorylates other target molecules, turning their proteins off or on. Protein kinase A can also enter the nucleus to activate transcription factors. Calcium can also serve as a second messenger in other common G protein coupled receptor systems. It achieves this using PIP2. It starts with a signal molecule binding to GPCR on the outside of the cell. The active G protein then activates phospholipase C. Phospholipase C hydrolyzes PIP2 into 2 other molecules. One is IP3, which also serves as a second messenger, and DAG, which remains in the membrane and activates protein kinase A. Protein kinase C phosphorylates the calcium ion channels, and it allows calcium into the cell. Calcium binds to PKC, and the protein comodulin makes it more active. Active comodulin activates kinase, which then phosphorylates target molecules that go on to do other things. The cell cycle is an organized process of cell division in life. It is crucial that nothing goes wrong during the cycle. Things happen at specific times and during specific phases. There are 4 stages to the cell cycle: G1, S, G2, and M. M phase is when cell division occurs, or mitosis. S phase is when DNA replication occurs. G1 and G2 are checkpoints that the cell uses to run diagnostics. This occurs to make sure the cell cycle is going right. Interphase is comprised of stages G1, S, and G2. This is when preparation for mitosis occurs. There is a cell cycle control system that ensures everything is running smoothly. Certain enzymes control the process of the cycle. Key proteins responsible for all the major events of each stage are alternatively turned on and off in the correct pattern to achieve the correct cellular effect. The primary mode of cyclic regulation of proteins during the cell cycle is phosphorylation and dephosphorylation. This occurs through the use of kinases and phosphates. The activity of kinases is staggered with them being activated at different points in the cell cycle. This allows the kinases to trigger the cell's progression through the cell cycle. Cyclins are proteins that control the kinases, and the kinases control the proteins involved in the cell cycle. It's foolproof regulation. Cyclins bind to and activate kinases. Because of this, kinases are collectively called cyclin-dependent kinases, or CDKs. There is a cyclin that binds to CDK specifically for S phase, G1/S phase, and G2/M phase. With the cyclin bound to CDK, the cell cycle can continue. The cyclins and CDKs are specific too. They can only bind with their corresponding molecule, and each cyclin CDK complex triggers different cellular events to prepare the cell for the next stage of the cell cycle. The cyclins can also be inhibited. CDK inhibitor proteins can stop the cell cycle at checkpoints. For example, DNA damage can be sensed in G1. That activates the expression of p53, a transcription factor that binds to DNA, allowing it to repair. These proteins block interactions between cyclin and CDKs, stopping CDKs from being activated, causing an arrest, and they won't be able to activate it until the damage is fixed. Cell junctions are the way cells connect to things, like each other. Gap junctions are private communication systems that exist between neighboring cells. Connexons are the key component to them. Connexons are transmembrane protein complexes. The complex is composed of 6 connexon subunits. There is one found on each cell membrane, and they meet between 2 cells and create a continuous channel across the 2 cell membranes. Small, inorganic, and hydrophilic molecules can freely diffuse between cells connected by a gap junction. These types of cells are metabolically, chemically, and electronically coupled. Tight junctions have one main function. They seal cells together so nothing can leak through in between them. A tight junction is formed by claudin proteins and not claudin proteins. They are arranged like a zipper. This arrangement makes a vacuum-like seal between the cells. Adherens junctions bind one epithelial cell to another. Their purpose is to provide strength against mechanical stressors the cell might face. They do this through the use of cadherin. This is a transmembrane protein. Cadherins in neighboring cells interact tightly, linking 2 cells together. The cadherins are connected to actin filaments within the molecule through linker proteins called alpha-ketinin and beta-ketinin. This causes the cytoskeleton of the 2 cells to link, and they are anchored intracellularly and extracellularly. This interaction also requires calcium ions. Desmosomes function the same as adherens junctions. They link epithelial cells together via the cytoskeleton and protect against mechanical stressors. Desmosomes also connected to cadherins, but the cadherins are connected to keratin, not actin filaments. Hemidesmosomes found in keratinocytes of the epidermis of the skin link to collagen using integrins. They anchor the epithelial cells to the basal lamina through intermediate filaments. The occurrence of cancer is defined as when a damaged cell enters the cell cycle and divides, passing the cellular instability onto daughter cells. It has 2 main properties. The first is that they proliferate uncontrollably. The second is that they invade and colonize regions of the body that are reserved for other cell types. Cancer is caused by an accumulation of very specific mutations that come together in the unique combination that allows cancer to progress. Cancer is a disease of old age because a sufficient amount of time has passed and a sufficient amount of combinations have occurred. There are 3 types of cancer. The first is benign cancer. This is when a tumor is growing, but the cells remain localized in a specific area of the body. By this I mean the tumor can grow larger, but it will not move to any other parts of the body. So technically, a benign tumor is not cancer because it is not invasive. This type of cancer is the most curable. Usually, the tumor can be fully removed in surgery. The second type of cancer is malignant. This type of tumor not only grows in a localized area, but it has the ability to invade surrounding tissue. Like said, cancer is invasive and it wants to spread and spread. The third type of cancer is metastatic. This type of cancer is when the tumor cells not only invade neighboring tissue, but they have invaded a blood or lymph supply. This causes a secondary tumor, meaning the cells have migrated from the original site to a different part of the body and begun growing there. Cancer can easily be described as sociopathic. A sociopath is someone without a conscience. Cancer is just that. It only cares about its own cells surviving and passing the damaged DNA onto daughter cells.